In this lecture, we are going to discuss non-polar amino acids. Basically, different proteins actually made up of amino acids, and different amino acids combined through the bond or linkage that is actually called the peptide linkage, or you can say peptide bond. Okay, so if you see here, four amino acids 1, amino acid 2, amino acid 3, amino acid 4. Here are basically the four amino acids that are actually combined by the peptide bond, or you can say peptide linkage. Okay, so here is the peptide bond 1, here is the peptide bond 2, here is the peptide bond 3. So if you notice that number of amino acids actually 4 and the number of peptide bond is actually 3. So it's mean that in proteins, you will see the number of amino acids will be 1 larger than the number of peptide bond, or you can say peptide linkage. That's the most important thing. Peptide bond will be 1 less than the number of the amino acids. Okay. If we look at the types of the amino acids, so basically you will see two major types of the amino acids: non-polar amino acids, non polar amino acids, and the polar amino acids. Okay, here are basically the two major types of amino acids, and the polar amino acid further categorized into the two, that will be the uncharged, having no charge, and the charged amino acids. Charged amino acid and the uncharged amino acid, that is actually lie in the polar amino acids. Okay, if we discuss the different conditions of the polar and the non-polar amino acid, before we discuss, you should know that the blue color will represent the polar amino acids and the red color will represent the non-polar amino acids. We will discuss in two situations. First one is the hydrophilic situation, hydrophilic, and second condition will be the hydrophobic conditions. Okay, if we put the amino acid in the hydrophilic environment, water loving environment, then what will happen? So amino acids will change their arrangement in the hydrophilic environment. So in that case, polar amino acid will expose to the outside. Here are blue color will represent the polar. So all the polar amino acid will expose to the outside, and the non-polar amino acid will be buried inside. Non-polar amino acid will be buried inside. So that will help in the stability of the structures of the different proteins. In the hydrophilic environment, you will see the outer will be the polar amino acid because they can interact with the hydrophilic environment, and the non-polar will buried inside the polar amino acid. On the opposite side, you will see in the hydrophobic environment, water hating environment, in that situation you will see the non-polar amino acid will expose outside because they can easily interact with the hydrophobic environment, or you can say the polar amino acid will be buried inside. So here are basically the two structure in the hydrophilic and the hydrophobic environment, that how different amino acids arrange themselves in the different environments. Okay, but the question is, where does this polarity comes to the amino acids? So in this lecture we will see that how non-polar amino acid get non-polarity from which function group. Okay, so if you see 9 most important non-polar amino acids. Okay, so in that case you will see 3 group will be common to all. First one is the carboxylate group. Okay, that is the carboxylate group, and next group is the amino group, and third one is the hydrogen group. So basically carboxylate group, amino group, hydrogen group will be common to all amino acid. Okay, but the group that is different in all will be the R group. So in case of glycine, you will see the presence of hydrogen at the side chain. Okay, and the hydrogen is actually non-polar in nature. Okay, that's why glycine is actually the non-polar amino acid. It will not interact to the other amino acids. Okay. Second is the alanine. So alanine carry the methyl group at side chain. Other will remain the same, that is the carboxylate group, amino group and the hydrogen group. Difference will be at the methyl group, and methyl is actually non-polar in nature. Okay. Similarly, in case of valine, you will see the presence of two chain carbon, and at the first carbon you will see the attachment of the methyl group. So again, now this structure is also the non-polar. So that's why valine is also the non-polar amino acid. Okay, so it's mean that side chain will decide the whether the amino acid will be polar or not. Okay, in the leucine you will see the presence of the three carbon chain. Okay, and at the second last carbon you will see the presence of the one methyl group. That is the structures of the leucine. Again, now this composition is again non-polar. In the isoleucine, what's the difference you will see between the leucine and the isoleucine is the presence of the methyl group. In the leucine, methyl group is actually present at the second last carbon. But in that case, the methyl group will be present at the first carbon. That is the difference between leucine and isoleucine. In the methionine, you will see the three carbon chain. But between the last carbon and the second last carbon, you will see the presence of the sulfur group. That's the most important point about the methionine, presence of the sulfur atom. Okay. In phenylalanine, you will see presence of one carbon, and that carbon will attach to the benzene ring. In the phenylalanine, you will see the presence of the benzene ring. Okay. Tryptophan, you will see the one carbon that is attached to the ring structure, and ring structure is further attached to the benzene ring. So that is the structure of the tryptophan. Again, that is the non-polar in nature. Okay. Proline, you will see the presence of three carbon chain, that is one carbon, two carbon and the three carbon. So last carbon will again directly attach to the amino group. That is the exceptional case in the non-polar amino acid, because in that case amino group directly attached to the two carbon atom. So that's why it is the secondary group or secondary structures of the amino group. In all these situations, you will see the amino group will directly attach to the one carbon atom. You just have to remember, if the amino group, here is the NH2, directly attached to the one carbon atom, then it is called the primary structure. If the amino group NH directly attached to the two carbon atoms, then it is called the secondary. And if the amino group, here is the amino group, directly attached to the three carbon atoms, then it will be the tertiary structure. So in that case, in the proline, you will see the amino group will be directly attached to the two carbon. That is actually the secondary natures of the amino group. In all other cases, you will see the primary amino group. So that's the basic difference between the proline and the all others non-polar amino acids. So hope so you got the point. If you still have any question regarding the non-polar amino acid, and that is the non-polarity is determined by the natures of the R group, and in non-polar amino acid the R group will be unreactive. Okay, so this is all about the non-polar amino acid. If you still have any question, you may ask in the comment section. Thank you so much.